Welcome to Spring LIB.
Okay, so in 66, we're sort of coming up with a question we could use to further our studies. And the first thing to note is when we're comparing the Archaean methanogens, right? So we're looking at some archaea. We're going to want to compare it to the non-methanogenetic archaea, right? So you want to compare these two different kinds of archaea. And the reason you wouldn't compare it to a bacteria is because the bacteria is a whole nother species. So they're going to have lots of differences, and those differences might not be attributable to what you're searching for. Okay, so the thing to note is that these methanogens, right, they do this thing where they utilize carbon dioxide. Okay, and you'll see earlier in the textbook, it talked about reducing carbon dioxide. And for these terms to be reduced means to lose oxygen, and to be oxidized, okay, is to gain oxygen. That sort of makes sense when we think about it a little bit. And in this process that these methanogens go through, if we look at the chemical equation, right, so they are utilizing the carbon dioxide and then also some hydrogen molecules, right? And the result here is going to be some methane as well as some water. Okay, so what happens here is the carbon was reduced, right? Because it lost oxygen, but the hydrogen has been oxidized. It gained oxygen. Okay, so what we want to do is we want to look at the archaea structure from the non-methanogenetic archaea to the methanogenetic archaea and compare their structures and see if one or more of those structural differences leads to the ability to use this carbon dioxide to oxidize this hydrogen molecule, right? Because the hydrogen molecule gets oxidized here. Okay, so the correct answer is C.